So hello everyone, welcome back in my YouTube channel. So today I'm here to discuss a short topic named Kingdom Monera. Essentially, Monera is biological kingdom that is made up of prokaryotes, particularly bacteria, and they have not true nucleus. Such is composed of single cell organisms that lacks true nucleus. As for DNA is nagged are not bound by nuclear membrane, and it lacks organelles like mitochondria, lysosomes, plastid, and Golgi bodies, endoplasmic reticulum, centrosome. So in the Kingdom Monera, we are going to discuss Archaebacteria, Eubacteria, and gram-positive and negative bacteria. So without wasting of time, let's start. Monera is the first kingdom of all the 5 kingdoms. As well, they are unicellular, means single-celled, and they are prokaryotic and they are also microscopic. If we discuss about the mode of nutrition in the moneras, maybe autotrophic and heterotrophic. Autotrophic means they make their own food, and heterotrophic consume their food by other organisms to obtain energy. If we see the Kingdom Moneras, divided into 2 major groups. First, Archaebacteria, and second, Eubacteria. If we see, the Archaebacteria are usually found in extreme environmental condition like high temperature, low pH, high salt concentration, etc. Now let's see example of Archaebacteria. Number 1, methanogens. Methanogens present in the gut of several ruminants such as cows, buffaloes, and they are responsible for the production of methane biogas from the dung of these animals. And they live under anaerobic conditions. Anaerobic conditions means where lack of oxygen. I have explained you the gut of ruminant animals like cows, buffaloes. Now see second examples is halophils. They live under extremely saline or salty concentration. Example 3 is thermoacidophils. They live under extremely hot and acidic conditions such as hot spring. So finally we have done with Archaebacteria. So let's see Eubacteria. Eubacteria is also known as true bacteria. They are advanced as compared to Archaebacteria, and they are characterized by the presence of rhizate cell wall, which is made up of peptidoglycan, and there is presence of flagella if motile. Motile means Eubacteria can move due to the presence of flagella. Now if we discuss types of bacteria, so number 1 is Cyanobacteria, also refers to blue-green algae. They have chlorophyll A similar to the green plants. They are photosynthetic autotrophs, and they are unicellular, colonial, and filamentous as well, and they form algal blooms in the polluted water bodies. The most important point is some of them can fix atmospheric nitrogen in specialized cells called heterocyst, as in nostoc and abina. So next, second example is chemoautotrophs. They use inorganic chemical substance as nitrates, nitrites, ammonia to produce energy in the form of ATP, and they play a great role recycling nutrients like nitrogen, phosphorus, iron, sulfur. The next example, heterotrophic bacteria. Heterotrophic bacteria are most abundant in the nature, and many of them have significant impact on human, like help in making curd from the milk, production of antibiotics, fixing nitrogen in legumes, root, etc. And some are pathogens causing damage to human beings, crops, farm animals, and pets, like cholera, typhoid, tetanus, chytris, canker, as well non-disease caused by different bacteria. And if we discuss the reproduction, bacteria reproduce mainly by the fission, and sometimes under unfavorable conditions they produce a spore. And they also reproduce by the sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacteria to other. So next point is mycoplasma, PPLO. Mycoplasma are the smallest living cell without cell wall, and they can survive without oxygen. The most important thing is many mycoplasma are pathogenic in the animals and plants. If you see, the pathogenic means disease causing. And if you will observe the bacteria, there are different shapes of bacteria like coccus in the spherical shape, vibrio in the comma shape, bacillus in the rod shape, spirilla in the spiral shape. So our next topic is gram-positive and gram-negative bacteria. So there are 2 different types of bacteria, as well cell classified according to the difference in the cell envelope. And these bacteria are developed by the scientist named as Christian Gram. He developed a stain and named it crystal violet because its color is also violet. He took some bacteria and put them in the stain, and after some time he observed some bacteria that take up stain and turned purple color, named gram-positive bacteria, and some bacteria that did not take stain and remain colorless, named as gram-negative bacteria. So if you have any doubt related to this video, then you can comment below, and please like, subscribe and share this video with your friends, relatives so that they can also get help from this video. Thank you so much.